Welcome to Unit 2. In this unit, we're going to be learning about the cell and everything from cell compartmentalization all the way through to endosymbiosis. So please remember to ask if you have any questions and come join our community on Skool.com for extra resources. Now, the guiding question here is how do transport proteins help substances move without ATP? So we're going to learn about ion channels. We're going to learn about transport proteins, which are also referred to as carriers. And then we're going to learn about aquaporins. So you need to be able to explain how all three of these type of proteins facilitate diffusion using facilitated diffusion down the concentration gradient with no ATP required. So why some substances require proteins? If we look at substances that are charged, okay, they cannot pass this non-polar region or this hydrophobic region. They cannot pass it. So because they have that charge, they cannot cross that hydrophobic region. And if they're polar and large, they can also not cross this non-polar layer. Okay. So facilitated diffusion, just a recap. Facilitated diffusion is we move from a high concentration all the way to a low concentration. And it's called facilitated diffusion because there is a protein involved in assisting the molecule from moving from a high to a low concentration. But very important, no ATP is required because we move down the concentration gradients. Now, ion channels, okay? Ions do not move easily through this hydrophobic membrane interior. So this hydrophobic interior, ions cannot cross it because they are charged. Now they can either be positively charged or they can be negatively charged, but as soon as they are charged, they cannot cross this cell membrane. They need a transport protein or a channel. So ion channels allow specific ions to pass through. So it's very specific. So this protein channel can, for example, just pass through potassium or just sodium or just chlorine. It is very specific in the ions that can move through this protein. Now, large polar molecules. Large polar molecules, like glucose, for example, they're big, they are polar, and they have no charge. But because they are big and polar, they need a protein, and they specifically use carrier proteins. Now, carrier proteins, the molecule will bind to the protein, and once the protein binds, the shape of this protein changes. And because the shape changes, it releases the glucose into the cell. And once this glucose is released, it will resume this normal shape. Okay. Now, it is also done by facilitated diffusion when they move, because they move down a concentration gradient. They move from high to a low concentration, okay? And it happens through a protein, but there is no direct energy input needed. It is once this molecule of glucose binds to the protein, the shape of the protein changes. And because the shape changes, it releases it into the cell. And once it releases it, it resumes its normal shape. So it's like connecting, releasing, connecting, releasing. but it does not require energy. Aquaporins. And I'm going to use a lovely blue for this one. Aquaporins. Aqua means water, and porins are these like protein channels that are specifically for water. They transport large quantities of water. Now remember, water is a small polar molecule, but it has no charge. It can move slowly through the membrane, okay? But if I want to get water quickly into the cell, I use an aquaporin. And they are specialized channels that allow water to pass quickly. If you enjoyed today's lesson, please don't forget to like and subscribe, and come join us on the Skool.com community page, where you'll find AP Bio blueprints. And if you take a look inside, you will find that Unit 1 is completely free. See you there.